Well, I'll just make a start. Welcome to today's Research Translation Seminar Series. My name is Julia Reno with the TRI Communications team, and I'm just standing in for Professor Ranjani Thomas, who was hoping to chair this session but was unable to make it for the start. We're very privileged today to have with us Dr. Angus Forster and Mr. Blake Wills, respectively leading up in Australia Microba and Vaxxas, Vaxxas and Microba, pack the front. These two biotechs are just going at a phenomenal rate of growth and development. And we're so excited to have them based here at TRI with their R&D. And it's going to be really good to hear from them today about the milestones that they've been reaching in the last year, particularly with the limitations of COVID. It doesn't seem to have stopped them. And also the research collaboration opportunities that are available with these biotechs for our researchers based here at TRI and elsewhere. Before we begin, I'd just like to begin by acknowledging and paying my respects to both the Jaguar and people and the Turrbal people, the traditional custodians of the land on which we are gathered today. I would like to pay my respects to their elders, past, present and emerging, and acknowledge all Aboriginal and Torres Strait Islanders here today, both online and in person. And yes, to begin with, just because he was here in the building, in the auditorium first, we're going to start with Dr. Angus Forster, and he has worked in medical product development and commercialization for 20 years. His overall responsibility for research, development and operations of Vaxxas. And since 2012, he has established the Vaxxas R&D team to include now 64 scientists, engineers and professional staff, and led these into early stage clinical studies and many other things which we'll now hear. Thanks.
Fantastic. Thanks, Julia, for the introduction. It's great to be presenting again to the TRI community, especially on the topic of collaborations. We do a lot of collaborating, as any company working in research and development needs to do to remain successful, and it gets at the forefront of technology development. So I'm going to spend about 20 to 25 minutes just talking about Vaxxas in general terms, not too much about our sort of project development milestones, but more about the company and the areas of technology and research and development that we do. But I thought I'd start with a first slide which really shows our team. And as Julia really nicely put it, we've been very, very fortunate in the last sort of 12 months to have almost doubled in size here at the TRI. So we're around about, I think about 64 full-time researchers working in Vaxxas at the moment. This is a photograph from our Christmas party at the end of last year down at the Howard Smith Wharves. And we were so fortunate, I think, as a Brisbane-based company to be able to get together and have a Christmas party and socialise as a company. But it also, when we look back at the previous photographs from other Christmas events that we've had, just the sheer number of people that we've got now in Vaxxas that have joined in the last 12 months in itself was a tremendous story of company growth and sort of evolution of what we're working on. And we work in a whole different range of areas. We're working hard to take our product through various clinical studies, but also trying to build up the capacity and capability to manufacture products at high volume and at low cost. And the other thing that sort of makes you recognise is that in this team of sort of 60 people, there's a real diverse bunch of scientists, engineers, quality professionals, a smattering of business development, preclinical scientists, even a few people that specialise in working with various different types of animals. So our team is very, very diverse. And as a company grows, and I think Blake will talk about Microba a little bit, you also need to think about what your company stands for. So at the end of last year, as we were growing, we thought we needed to put in place some sort of value statements for what our company stands for as a community, if you like, as we expand and grow. So we had a sort of couple of group interactive meetings. We put our heads together and thought, well, things that were important to us as Vaxxas are sort of making a difference. So we think our technology can make a difference to vaccination, both in situation of pandemic response, but also, you know, in low and middle income countries where access to vaccine can be very, very challenging. I think we work at the forefront of technology, you know, research and development in a number of different areas, and I'll show you some examples as I go through some of those sort of areas of focus for the company in our research and development. So we need to be thinking about what's coming next. So sort of leading the way in terms of our technology development, the experiments that we perform, and our ability to look forward to where we need to be in sort of 5 years' time. And I think importantly for today, it's about, you know, how do we collaborate internally as a company across this diverse set of individuals working in Vaxxas, but also how do we collaborate with other groups, companies, regulators, and the community as well, to make sure that we're accessing everything that we need to access, but also at times also, you know, giving back as well in terms of, you know, the progress that we've made and how we can work with the greater community. For those of you that don't know us, we're in a UQ-based spin-out, so formed in 2011. I joined in 2012. I was fortunate enough to be the first Vaxxas employee, and I've managed to cling on, you know, for almost 10 years and not get turfed out. So, and I've been sort of there since the start of our sort of journey, which has been really, really neat. So we've been based in Brisbane since the technology came out of the University of Queensland. Now we're privately funded. Our main investor is One Ventures based down in Sydney, also Brandon Capital Partners in Melbourne, and then an overseas investment company, US Healthcare Ventures as well. So we're a privately funded company. So what are we doing in Vaxxas? So now what's wrong with the needle and syringe for vaccination? I guess, you know, a few of you online and a few of the audience would have maybe run down the stairs last Friday to go across to the PA hospital and get your COVID-19 vaccination. And it would have been given with a needle and syringe, which is a very effective way of giving a vaccine. And it's been around for 100 years. But we think there's a better way of, you know, giving vaccines, which can overcome some of the challenges and limitations that you've got from needle and syringe. And I think now what's been interesting in the last 18 months is the focus on development of new vaccines for COVID-19, which is entirely appropriate. The world is desperate for a range of different vaccines, which can help us get over this huge issue that we're facing as a global community. But now the sort of focus has shifted to how do we roll that out? How do we vaccinate all the people that we need to with the vaccines that are increasingly becoming available? And the needle and syringe has certain limitations. You need to be trained to use it. You need a nurse or a doctor to administer the vaccine. Especially with the COVID-19 vaccines, they need to be stored at a very low temperature, you know, -70, -20 with the mRNA vaccines, or potentially 2 to 8 degrees with the other sort of other vaccines. The response can be slow in terms of building up effective protection from the vaccine and require more than one dose occasionally to provide that protection. And also, you know, the dose you need to give that vaccine can be quite high as well. So that limits the number of doses you can make from the same amount of vaccine if you need a lot of that vaccine to generate those protective responses. But I think the biggest limitations are really around logistics and actual distribution. How do you get people vaccinated rapidly? How do you transport vaccines to where they need to be in Australia, but also more importantly in countries that have no cold chain sort of infrastructure set up, where that becomes increasingly challenging. So we think our technology can really overcome a lot of those challenges for a whole different range of vaccines. So we think we can make more doses of vaccine from the same amount of starting material, so increase, I guess, the availability of vaccines. We've designed a technology which is simple to use to vaccinate, so suitable for lower trained individuals to vaccinate, or potentially in certain situations for rollout to homes and for self-administration or administration from a guardian or family member. We think by delivering vaccine into the skin and targeting the immune cells there, we can improve the immune response you get with vaccines as well. So potentially using less vaccine, or maybe enhancing the sort of onset of protection from those vaccines. And also importantly, and I think particularly in the case of you know, the COVID-19 vaccines that are stored at such low temperature, the ability to improve the stability of the vaccine to higher temperature distribution, so improve its thermostability, is another potential advantage of our technology. And just to play a video, this is the concept. So this little application system, I've got one of my, you know, up here as well if you want to take a look later on. It's applied to the skin. It's actuated, as you saw. It's left on the skin for 10 seconds. It's removed and discarded. So that's the process of vaccination. So, you know, very, very simple. No parts to assemble. It's supplied, ready to use. The actual patient receiving the vaccine does not see the patch which actually delivers the vaccine into the skin. So the whole thing's been designed to be low cost, simple to use, and entirely disposable. How does it work? In the center of that device, and if you come out and have a look, you see it in the middle of this product, there's a small patch. That small patch, which is made of plastic, is covered in this very dense array of small micro projections. So we call it a high density microarray patch. Those projections can barely be seen with the naked eye. They're about 250 microns in length. And we have around about 1,600 small micro projections across the sort of the patch you can see in that photograph on the bottom right. The way we put the vaccine onto those plastic micro projections is by dispensing controlled amounts of liquid onto each of those projections very, very rapidly, and then drying that solution quickly in place. And you can see in the top left-hand corner is a scanning electron micrograph just showing the coating of vaccine formulation on the tips of those micro projections. It needs to be up there because when it goes into the skin, we need to deliver that vaccine effectively into those top layers of the and target those immune cells. So why do we do this? I guess, skin vaccination has been around for a long time. There's nothing new with skin vaccination. It's a well-known route of vaccine administration. I just think the embodiment of the technology to do it has been very, very challenging. The skin is a repository of an abundant source of immune cells, especially in the epidermis and the upper dermis. So that's where we target delivery of the vaccine into. We're targeting those immune cells in those top layers. The way the product works is a bit of damage done at the site of application with the patch. You're putting 1,600 small holes in the top layer of the skin to deliver the vaccine. And that sort of local damage creates a few alarm signals at the point of vaccination as well. So the point you're delivering the vaccine into those immune cells, you're also triggering their immune response to take up that vaccine and sort of move it to the lymph nodes. So we think that can boost immune responses, and I'll show you one or two slides on some of the results we've got from clinical studies which show that sort of dose-sparing potential of our technology. Who do we work with? And I guess in the spirit of collaboration, we don't develop new vaccines. We don't research ways of looking at immune responses. We collaborate with partners who have novel vaccines or vaccines that they'd like to change the delivery routes of and who want to collaborate with us. So at the moment, we're working with the Gates Foundation on measles and rubella vaccine for low and middle income countries. We've been working with the Gates Foundation for, I think, 5 years now. We're also working with Merck Sharp and Dohme, one of the world's largest vaccine companies. I think the largest company by revenue in vaccine sales. Well, they were until maybe COVID came along, but certainly one of the premier vaccine companies in the world. And late last year, we announced a relationship with the US government, so the Biomedical Advanced Research and Development Agency, looking at pre-pandemic influenza vaccination. So even with COVID going on, the US government was thinking, well, how do we prepare for the next pandemic and come up with better ways of delivering vaccine to the US population? So that's a funded program right through a large phase 1 study with an influenza vaccine delivered through our patch technology into healthy subjects. But we're also interested in other vaccine candidates and also immuno-oncology targets as well. So we are looking at building up our pipeline of different vaccines that come into Vaxxas. We test their capability and compatibility with our patch technology and then move them through the development sort of pathway. That's who we're working with. So we're, by virtue of what we do, we have to collaborate with a range of different partners. And these partners are very demanding: Merck, BARDA, US government, and also the Gates Foundation. We have to make sure we're delivering on the milestones associated with those projects and meeting their requirements. Just some immunology. We've done a number of clinical studies. This was our most recent back in 2018, 2019. This publication came out in 2020, looking at a single monovalent H1N1 influenza vaccine delivered by our patch technology. And this plot shows the functional immune response to application of our patch to the skin at 2 different sites. So the upper arm site, UA, and also the forearm site. And this shows a response to a dose which is similar to that given by a needle and syringe. So the patch results are shown in blue and red. And this is a small study, 20 subjects per group. And typically in small flu studies, you don't expect to see significant differences or differences between groups, just given the nature of the immune response that you see in the variability of that. So we were really happy with this data, showing that up to 60 days post-administration, that our functional antibody titers were statistically higher than that at the same dose given by a needle and syringe. That was a good outcome. We then thought, well, let's go look. We also went down to lower doses as well. So this shows, 1/6 of that dose given by needle and syringe, administered by the patch to the same number of 20 subjects per group. And essentially what we saw at a 2.5 micrograms of active, which is compared to 15 micrograms via needle and syringe, our responses essentially overlaid and matched what we saw with needle and syringe. So this is a great outcome. And what this means is that potentially you can give a lower dose of vaccine and therefore release more doses to the market more rapidly. And that's what that very complicated table shows on the right-hand side. If you can increase vaccine output, you can essentially save the economies of the world an awful lot of money in the current sort of pandemic situation. Of course, you've got to be able to manufacture, and that's one of the other challenges. Thermostability, same study, same vaccine. We also looked at whether when we actually do store the vaccine for a period of time at different temperatures, does it remain stable? And we also compared that to the standard liquid vaccine, Fluria Quad, containing the same antigen. So we stored our product for up to 12 months at 40 degrees, and we measured its potency over that time and saw no change in the potency of that vaccine when dried onto the patch. So if you compare that to the Fluria Quad, the liquid vaccine, you store that at 40 degrees for 1 month and you get a significant reduction in potency. So this just shows there's an opportunity with drying the vaccine and presenting it in the way we do to massively improve the thermostability. What else do we do? We've done usability is a key aspect of what we do, and also looking at what impact a new vaccine delivery technology can make on vaccine logistics. So a while ago now, we ran a study with the WHO, World Health Organization, without a patch, purely a usability study in 3 countries, Benin, Nepal, and Vietnam, looking at whether people could use the technology when given simple instruction about how to apply it. And that went very, very well in this context, right, in terms of going to a non-native English-speaking community with a simple instruction for use and giving this to a range of different users. So trained healthcare professionals, but right down to sort of lower skilled community healthcare workers as well. And we can see that there was great feedback from us with the usability of our product. So we run usability studies. As I mentioned, we work on manufacturing scale-up and have to come up with new technologies which enable us to make this product at high volumes and low cost, but also very, very rapidly, and have all the controls in place that are needed to take a regulated product through to market. So we have a big engineering contingent in Vaxxas that work on a whole range of different technologies. And this is just the way we dispense the vaccine onto the microprojection. It's essentially an inkjet printing-like process, but happens with much more control and also dispenses many, many drops at once to make sure we've got the throughput. And that's technology that we've built, researched, developed, and constructed at Vaxxas. Some of you may be familiar, at the TRI here, we make product for clinical studies. So phase 1 studies, we've made at least 4 or 5 different batches of product for various clinical programs. And those products are made by our team in the building under controlled conditions in the clean rooms at the TRI. So we do have people coming in from the TRI community and working in those clean rooms occasionally and gaining some experience in the quality management systems that are needed to take products through to early stage phase 1 manufacture. Because it's a vaccine, can't be terminally sterilized, it has to be made under very, very clean conditions, so aseptic manufacture. So that puts quite stringent requirements on the operators and the facility in which we do that work, which is sort of shown in that photograph. Conscious I'm going to take up Blake's time, so I better move on. So what are we doing? So what are we doing next? We are moving to a new facility. We have a collaboration with the Queensland State Government to build the next facility for Vaxxas, and that's been built in Brisbane. It's going into the North Shore area. Hopefully won't be subsumed by the Olympic Games when that comes to Brisbane. So it's right in that space there. So the Queensland Government is helping us with the build of a new facility, which we hope to move into sort of midway through next year. And that's designed to enable us to produce product for late stage clinical studies. So that to do that, you need a manufacturing license from the Therapeutic Goods Administration to work to certain quality standards. So the whole facility needs to be designed to enable that late stage clinical trial supply and also accommodate our growing team and laboratory space and things like that. So that's a transition that we're going to be making. It's sort of on track, and the schematics are in for the design and build. And hopefully it's going to be around 12 months before that facility is in place. So that will allow us to expand out, particularly in our manufacturing capability at Vaxxas. So kind of what do we look to in the future? And this is, as we move through different phases of clinical development, you can see the way the company starts to grow and expand. And a key part of it is trying to keep, I guess, the technology and also the company doing that work, ideally here in Queensland and in Brisbane. So obviously building out our R&D capability and maintaining that here in Brisbane to support a growing number of vaccine programs through preclinical development, right through early stage clinical development into sort of late stage clinical. And that employs a lot of graduates from various institutes around the Queensland area. We have to make the device. So it's a patch and an applicator. Those products need to be manufactured. So to have that capability to do that manufacture ourselves, which is very different to the vaccine side. It's big machines, smashing metal, injection molding of plastic. To have that capability to apply those devices either into our own vaccine manufacturing plant or to other collaborators that can then make product is a key thing that we're trying to build this facility to enable that work to happen here. And then, as you move through to late stage clinical, your demand for control of process and throughput of process goes up significantly. So the range of manufacturing technologies that you need grows sort of exponentially through development. So we're also working on having a finished product manufacturing line here in Queensland, which can make product for phase 3 studies, but potentially for some commercial, early stage commercial supply, maybe in the Asia region, but also potentially to help potentially make product for future pandemics here in Australia. And obviously sovereign manufacturing capacity is a huge topic in the Australian government at the moment. So that's what we're working toward. And I just want to end with a few slides talking about the nature of the collaborations that we do. And I said, I think we collaborate in a lot of different ways. We access various equipment, either here in the building or with other partners. The fact that we're in the TRI in itself is a collaboration. We access a lot of different facilities within the TRI, the vivarium, the clean rooms, working with other researchers, the CRF across the road. So we already, by virtue of being here, collaborate very broadly with the TRI community. We also work with AMP as well, significantly on the device manufacturing side. We take placements. So at the moment, I think we have about 4 placements. So one of them is right here. So it's Alex Owen from QUT, undergraduate. We also have a postdoc, Ilka Hacker from QUT. We have Mega Budwani from the Fraser Lab. She's doing a CEF Metcalf Fellowship for 3 years in Vaxxas. And we also have Jenny Zhang from the Wells Lab, who's just started a PhD placement at Vaxxas. So we're a bit busy with placements at the moment. We've got four going on at the same time, but that's great. That gives people an opportunity to work at Vaxxas for different periods of time. Grants, we do work with grant programs. So we've got an IMCRC grant with the University of Sydney, looking at usability clinical studies. And we've had that grant sort of ongoing for a couple of years. And we also work with Dr. David Muller across at UQ, who's doing his advanced research at Queensland, a research fellowship looking at COVID vaccines on our patch technology as well, which is great. With new vaccines, novel vaccine candidates, if people have interesting immuno-oncology or vaccine candidates, I'd like to see if they could benefit from being delivered through the patch technology. We often work on a sort of no cost basis. We get the vaccine, we take the vaccine, we run the preclinical programs, see if there's a benefit. So we do that with, we're doing that with Griffith University, been working with them for a while on Group A Streptococcus. We also have a collaboration about to start with the University of Turin in Italy as well. Been a bit slow with some of the COVID challenges they've had in Italy. And so that's why we like to work with either companies or academic groups who have novel vaccines or immuno-oncology compounds that we'd like to take onto the patch. We do offer part-time work experience, especially in our clean rooms. We'll get to allow people to get insight into good manufacturing practice and working in a manufacturing environment. So I think we have Pumi working with us at the moment, 2 days a week, helping out in our clean rooms. She's also from the TRI community. Consultancy for academics and experts in the area. We have a whole range of different consultancy arrangements in place, key opinion leaders, technical advice on what we're doing, especially immunology. And then we do fund external projects. So there's an area or topic that we're interested in working on. We will fund that research and development with either industry partners, contract manufacturers, contract development companies, or potentially academic labs as well. So we've worked with the University of Kansas for 3 or 4 years on quite large outsourced vaccine formulation programs, for example, which we pay for, and obviously it's with a contract piece of work. So the types of collaborations that we have, and then what do we what are we interested in? I mean, the topics are pretty much endless. I think anything to do with our technology. So obviously new vaccines, as I've mentioned, why our technology works the way it does in the skin. So looking at skin immunology, looking at novel adjuvants or existing adjuvants to see whether they can improve the immune responses and enhance the, the effects that we get. Material science, there's obviously a patch and a liquid that goes onto it. So understanding the material properties, surface interfaces, and those types of things is important. Vaccine formulation and analytical equipment. So novel ways of determining what we've done when we've dried the vaccine onto the patch and released it. Manufacturing technology, all our manufacturing scale-up is essentially done in collaboration with a group in Germany called Harry Hoflinger. They're one of the world's largest pharmaceutical automation specialists. But there's a whole other thing associated with analytics, vision systems, etc. So we sort of have a broad set of partners that we work with on manufacturing. And then other things as well, as I said, human factors, but also what difference can these products make in certain situations for vaccination? So especially in the pandemic. So what's the economic benefit and cost saving that you may get from this type of technology? And how does that then feed into public health policy and vaccination rollout? And we do clinical trials, and that's always done with external partners as well. And the opportunity to collaborate and work with the CRF across the way here as well is another area of opportunity. So yeah, that's what I wanted to sort of go through this afternoon, and I think Blake's going to come up next and go through the Microba story, but happy to take some questions at the end. Thank you.
Thank you, Angus. And yes, as Angus said, we'll be taking questions at the end, so please save them up. Now I have the privilege of introducing Blake Wills, who's the CEO of Microba. I've got a little blurb on you, which I hope is up to date. He's a proven company executive with experience in finance, life sciences, and education sectors. Prior to Microba, he was the chief operating officer of an ASX listed company. He brings substantial operational, financial, and governance experience to Microba as the founding CEO. His financial background, combined with his extensive operation expertise, has driven Microba forward as a thriving company with more than 50 full-time employees, probably more by now, but I'll leave that to Blake.
Fantastic. And the slides will roll over. There we are. Thank you very much, Julia, and thank you everyone for coming and listening to Microba today. It was a great presentation. We will touch on themes of collaboration as well. Clearly, that is the opportunity, trying to build a biotech company here from Queensland and take it globally, which is the marketplace we all compete in. So collaboration is actually a key tool in which we unlock some of that. Just skip forward and talk a little bit about what we do at Microba before getting onto collaboration. Clearly, the gut microbiome has been an enormous trend over the last decade, arguably even longer. And there's now some really compelling evidence demonstrating the strong links between the microbiome and disease. At Microba, we have a hypothesis that precision measurement of the human gut microbiome is required to solve large unmet medical problems. And we believe we have the leading technology to analyze the human gut microbiome. Why is there still so much opportunity in the gut microbiome if there's been so much focus on this area? The key is that the gut microbiome still really has not been unlocked. There was a paper a couple of years ago that Phil Hugenholtz, one of our co-founders, was a co-author on, demonstrating that 71% of the human gut microbiome has not been brought into pure culture. That means we haven't studied it in our traditional methods of culturing something in the lab and testing its physical properties. So at Microba, we're really focused on the entire gut microbiome, not just the cultured microbiome. And we've developed techniques to culture the unculturable. We also have very capable measurement technology. And to give a real example of that, using our shotgun metagenomic approach compared to the best-in-class alternative shotgun metagenomic approach, we've been able to identify more precisely and more comprehensively the constituents of the human gut microbiome. In a single US sample here, we talk about 183 species versus a competitor only identifying 81 species. So in a very simple example, you're missing half the picture if you're using the wrong technology to analyze the human gut microbiome. We've recently released a paper called Evaluation of the Microba Community Profiler, And if anyone's interested in digging into the actual scientific basis of our profiling technology, I'd strongly encourage you to take a look at that paper. To give a few real examples, if we use the MCP or the Microba Community Profiler versus Metaflan, which is the leading academic alternative that's broadly used through industry and academia, we are achieving substantially less false positives. We're achieving low false negatives, meaning that we're seeing a much bigger part of the community, and that overall it's a more comprehensive and accurate picture of the human gut microbiome. So what we've developed at Microba is end-to-end microbiome capability. We have an optimized laboratory process, which is managed here at the Translational Research Institute by Dr. Nicola Angel. She's done a fantastic job, and that includes everything from sample collection, which is a key part of microbiome analysis. If you don't stabilize a sample correctly, you're going to get community outgrowth and misrepresentation of the DNA that you're ultimately measuring. So we've optimized end-to-end our laboratory process. We've developed bioinformatic pipelines that we believe best in class that I've talked about, but we've also developed the data infrastructure that lets us do this securely and globally with some partners that we now have both in the US and Europe. Ultimately, this lets us deliver solutions. Those solutions may be for consumers, healthcare practitioners, or researchers that are trying to understand the microbiome in the context of the problem they're trying to solve. Why can we do this? Professor Phil Hugenholtz and Professor Jean Tyson, put your hand up in the audience for those who don't know Phil and Jean, have been leaders in the field of microbiome analysis for the last 20 and 30 years, respectively. They've published some of the seminal work in the field. Jean, whilst at Berkeley, published the first use of shotgun metagenomics to analyze a microbial community. They've developed some of the key tools that have been used over the last decade to analyze the human gut microbiome. Many of those are in fact open source. And Phil's been an absolute leader when it comes to defining the bacterial taxonomy. And he's recently done some fantastic work around a taxonomy called GTDB, which is getting an enormous amount of adoption. So Phil and Jean spent an enormous amount of time in the US before coming back to Australia in 2010 to set up a new research institute at the University of Queensland. They developed our core technology at Microba between 2010 and 2017. In 2017, we spun out of the University of Queensland and we started on our commercialization journey. And that's been an incredible journey for everyone involved. Unlike Angus, I was actually employee number 2 at Microba because I was in the office with employee number 1, who unfortunately got a grant that made him employee number 1 because we had the money to employ him. So I was employee number 2 at Microba. But since then, the team's grown enormously. We've clearly got the support of Professor Ian Fraser, who's our deputy chairman, chairs our medical advisory board and is very generous with his time. Associate Professor Lutz Kraus, known to many in the crowd as well. He's been promoted to our chief scientific officer. A big part of the Microba proposition is around using big data to interrogate the microbiome to define signals, biomarkers, and therapeutic candidates. And Lutz clearly has a fantastic skill set at that with his background in Switzerland at Nestle, also his work in cancer genomics, and generally speaking, being a leader in big data analysis. But the team's broadened out much further since then, and we've got a fantastic mix of excellent scientists, both here in Australia, but we've also built out our business development capabilities globally with a number of employees now in the US. So as a team, we're around 48 staff. That number gets outdated pretty regularly. We are growing. We have global operations. We have a distribution partnership in the US and Europe. We're negotiating a deal in the Middle East at the moment, and we've been supported through investment, which is a key part of being able to be a viable commercial entity who's developing new technologies. So what is our commercial model? How do we thrive and how do we work with others? We really have 3 components to our commercial model. At our core, microbiome analysis. We believe that we're the leaders in this space, and we believe that microbiome analysis services offered to consumers, clinicians, and researchers has a genuine value proposition that we can help those individuals or those groups understand the microbiome in the context of the problem they're trying to solve. Secondly, where we have the ability to, and this is all done on an ethics-approved research study, opt-in and de-identified data set, We use the big data, particularly from our consumer product where we have the rights to do so, to interrogate the microbiome deeply in the context of disease states that we're interested in. It's not only the disease metadata that's important there, it's all of the confounding factors that can come with the microbiome, whether that be diet or antibiotic usage, sleep, exercise. There's many confounding factors. So we need to be able to really cut through the noise to ultimately get to new products, because that's how we're going to make a positive impact on human health. So we use that large data. We call it a human-first data-driven approach to product discovery. If we talk about the commercial model and how that looks, there's really 4 key areas that we're playing in. In the top left is the microbiome analysis engine that Microba is most well known for. Again, this is the provision of services. One thing that we have identified is that we don't need to be the retailer of the product globally, but we can enable partners in offshore jurisdictions who have a captive audience of microbiome testers or potential test users to offer their microbiome test powered by Microba. So we think of it as the Intel chip model, where we're powering microbiome tests globally. In the top right, personalized consumer health. So it's estimated that that's a $35 billion industry that at the moment is based on some pretty weak science, generally speaking. So we have obviously large probiotic and prebiotic and other supplement companies that are trying to provide consumers with solutions based on their scientific knowledge base. Typically, those probiotics aren't derived from the human gut microbiome in the first place. They lack real clinical data, and there's no personalization as to who gets what product. And we know that the microbiome is fundamentally different between a large number of individuals. So if we can personalize our approach to supplementation of consumers based on their gut microbiome, we believe that's going to yield much better outcomes. And certainly these large companies are very motivated to do that. Thirdly, cancer treatment response. So the microbiome has now been established as a key variable in determining whether or not a patient will respond to immune checkpoint inhibitor therapy. So Microba is launching what we believe the largest melanoma-focused microbiome study led out of the US at Moffitt Cancer Center, but also here in Australia, where we're actually accumulating the data set to validate the signature that we believe is relevant for predicting whether or not someone will respond to an immune checkpoint inhibitor. The pilot data for that looks very good, and we're excited to take that project further. But ultimately, that's about moving someone from a responder, a non-responder, to a responder. So if we can shift the microbiome in a positive direction, that will obviously have a huge impact. These drugs, for those who don't know, they work in 20 to 40% of patients typically. They are very expensive, typically costing around $100,000 a year. And from a patient, a payer, and a practitioner perspective, it's a big problem to solve. Finally, we have a drug discovery platform. This is where we take a big data approach to identifying therapeutic candidates. As I mentioned before, we have a very exciting data set that we believe is one of the best microbiome data sets globally, that we can differentiate healthy and unhealthy individuals, remove confounding factors, identify signals, and pull out the microbes that we think are actually driving the response or are implicated in that disease state. To go into a little bit more detail, our consumer testing model, we are talking about fecal samples. So we're talking about dabbing a piece of toilet paper, stabilizing a sample that gets processed at a wet lab. That could be our wet lab here in Australia or one of our partner wet labs where we've transferred our protocols. That gets pushed to the cloud once it's sequenced, and we use our bioinformatic engine to process that data and return it back to the partner. Our most exciting partnership in this space is with SynLab, which is Europe's largest pathology company. That's launched in Spain through the COVID pandemic, which was quite amazing. We're the only innovation project they didn't drop. And we're excited that they're about to take that into 2 new markets in Europe. Second, personalized consumer health. We've announced a partnership with Unilever. We have a second confidential partner, and we may have a third one shortly based on current discussions. As I mentioned, this is a very big marketplace that needs not only better science to determine what the right products are, but personalization. And Microba is very well placed to deliver that personalization. One of the things, and talking about the data set that actually lets us drive that personalization, here's a little bit of insight to that data set. And I apologize, it's small, but It's not meant to be read in its entirety. But you can see in the graphs on the right-hand side, you can see clear distinctions in certain disease signatures. So we have depression, Parkinson's, Crohn's, obesity, type 2 diabetes. We have a range of factors that are driving a difference in the microbiome between the healthy group and the disease state. But importantly, on the left-hand side, we have a huge amount of metadata where we can control for confounding factors. So it gives us a really powerful tool to interrogate the microbiome and understand what's relevant in the context of disease. The cancer treatment response program I talked to, the study title is Orbit-M. We expect to be recruiting our first patient in July or August, led out of the Moffitt Cancer Center. And if we can not only identify who responds to immune checkpoint inhibitors, but if we can move that patient in a positive direction, that's something that we're very excited about in terms of making an impact in melanoma and lung cancer. But also there is commercially a significant opportunity to get that right. Some of our early work in this space, looking at IBD, inflammatory bowel disease, we've seen an area under the curve there of 0.97 in terms of being able to identify a patient with IBD based purely on their microbiome. We can further differentiate that into the two subtypes, major subtypes, Crohn's disease and ulcerative colitis. And we've also done some work around treatment response, as I said there, the immune checkpoint inhibitor work and the anti-integrin work in an IBD setting. Our fourth pillar is around drug discovery. Our flagship program here is inflammatory bowel disease. We've seen a great ability to identify the microbiome that's associated with inflammatory bowel disease. But importantly, we're now using that human-first data-driven approach to test these candidates in the wet lab. And the results we're getting in the wet lab confirm that our human-first data-driven approach are working. And we're excited to be going into human trials. Our target is mid-next year. The thesis that we have here around microbiome drug discovery, and to give people a better insight to the broader industry, in the early days, fecal microbiome transplant was showing some exciting results in a range of indications. And it does have some efficacy and it's exciting, but it's a very crude measure. So we know there's a signal there, we just don't know why it works. Then we saw out of that data era emerge where there was a random library screening approach to drug discovery. That's where people would try and culture whatever they could from the microbiome and actually test whether or not there was a therapeutic effect there. That was exciting and led to some successes. But where we're at now is a data-driven rational discovery phase. That's where we don't only focus on what we can culture, but we focus on the entire microbiome, take a big data approach and identify or test those microbes in the lab to see if they indeed deliver the therapeutic effect. So in terms of a pipeline, this is what it looks like. We start with big data, over 8,000 samples. I think that number's over 10,000 now. We validate that in US and EU cohorts, very rich metadata. We go through a data-driven lead identification step. Importantly, we have genome-directed isolation tools, which means that we can actually pull these microbes out of a sample and grow them in the lab for further testing. That's a large unsolved problem. And certainly Gene and Phil have done a lot of work in that area. And then we go into a pretty traditional drug discovery pipeline where we push it through a range of preclinical assays before candidate selection and looking to which one goes into a phase 1 human trial. Just to share some data for the audience here, this is one of our lead assets, which is a single strain live biotherapeutic. So it's a novel bacteria that we've identified in our big data set. We've been able to grow it in the lab and we've been able to test it in a range of preclinical models. This model here is a DSS-induced colitis model. So we are looking at results here where our positive control is prednisone, which is a corticosteroid that acts very strongly in the context of ulcerative colitis, but it comes with negative side effects. And what we can see through the histology, histopathological score, epithelial injury and inflammation score is that the Microba single strain live biotherapeutic over a treatment period is achieving the same or better results than prednisone. Now that's a fantastic result, validating for us that the human-first data-driven approach is indeed working. At Microba, we're also interested in the bioactive molecules being produced by these bacteria, and we have a program underway in collaboration with UQ, actually identifying those bioactives. In summary, I just want to talk, please do get in contact with us because the topic of collaboration is the spirit of this seminar. And I just want to name a few of the collaborators that we do have. So at Microba, we've worked extensively with UQ, which is where the company was founded from. Phil obviously still resides there at UQ, and we have a collaboration with Rob Capon where we're looking at bioactives from the microbiome. We do a range of work with QUT, in particular Tony Kenner, and looking at other models where we're trying to test our microbes in animal models to see what effect they're driving. We've worked with Griffith on a metabolomics effort. We've worked with the University of Newcastle as well. In addition to that, we have supported interns, PhD students, and I think collaboration is such a key theme of building any biotech business out of Australia. One of the things, given the audience, that I also wanted to mention is what we are able to do at Microba is, I guess, really get on with it and make an impact in human health without needing to wait for the next grant round. That's what, being a commercial company actually unlocks. I think everyone in the room and everyone online undoubtedly wants to make a positive impact on human health. And by becoming a commercial entity, you actually get to do that in a largely unrestrained environment. So I think that's been what's been exciting for me personally. I know for Jean and Phil, that's certainly been exciting as well, being able to see opportunities and go for it. And doing that in collaboration with others is really just going to accelerate the process. So with that, I'll conclude and look forward to the Q&A session.
Thank you, Blake. We're now just going to open it up to questions. Just for our online audience, I'll just repeat any questions through the microphone. Ranjani, did you want to do this actually? Just seen Professor Ranjani Thomas, who is the chair of our Research Translation Committee. So I'll let you do that.
Do you have any questions from the audience here? Yes, Scott.
So one for vaccine advice. We hear all about the milestones story, and replacing the microbiome guts on one level, but there's lots of other areas of the body where microbiome you see opportunities of replacing the microbiome in the mouth, the tool, the lung, other areas, what the challenges there.
Thanks, Scott, for the question. A little bit of feedback. Maybe I don't need it. Can I hit a bit of online?
Is that microphone on?
It's quite good feedback from it. Yeah, so I think mRNA vaccines, I've been working for a long time. I mean, I'm not an expert on the science behind them, but the two COVID candidates are both delivered using lipid nanoparticles, so they're the carrier for the mRNA to allow cellular uptake of those mRNAs and then transfection. So part of our challenge is to be able to take those lipid nanoparticles, dry them down, and then disperse them when they're delivered into the skin. So we've done a little bit of work in this area. We are looking at other COVID-19 candidates as well, see which are best suited to our patch technology for delivery and therefore sort of and compatibility. But we've done work with DNA plasmid vaccines as well, so we know we can get good responses in preclinical models. But DNA tends to be more stable than mRNA as well. So I think that there's a lot of other groups looking at microarray patch technology. Some have published with various COVID-19 vaccine candidates and also looked at delivering mRNA using patch technology. I think the challenge will be a little bit on the local response you get. You would have seen I guess there's a new England Journal of Medicine article looking at the response on the skin to especially second dose of the Pfizer vaccine, and you do get a fair bit of local reactogenicity, which could be driven in part by the lipid nanoparticle, which is used to deliver the mRNA. So that'll be interesting when you've got a skin-based delivery system such as ours. What type of skin response do you get? A lot of it comes down to formulation work. Can you keep the integrity of the lipid nanoparticle, redisperse that, maintain that encapsulation, or can you actually deliver mRNA in a different way, dispersed in an excipient matrix, for example? So that's some of the things that we're working on, and that's actually an area of interest for us in terms of collaborating with groups that have that expertise, either in lipid nanoparticle formulations to stabilization, or potentially in other approaches to delivering mRNA into cells through skin delivery vaccines.
Thanks, Scott. One of the key challenges for any company or research group is focus and taking an opportunity far enough. So for Microba, it is very tempting to do more than just the gut microbiome, and we certainly want to, but within the gut microbiome alone, there's probably a dozen opportunities which would all take 3 to 5 years and cost $10 million each. So there's also an element of purpose as a company and focus. So you need an identity as a company. But this is a great opportunity for collaborations as well, because our technology can be applied to those other environments, and we are working with some people around that. So I think for Microba, it's unlikely we're the leaders in that field, but we'd be very open to collaborations.
Bill.
So, your new production facility is going to be panel. So I'm just wondering what you both think about the idea of localizing biotechnology companies. In Brisbane, we're still very much both great examples. But do you see any value in trying to create a precinct or an ecosystem where I mean, TRI actually one for set aside for national activities, but do you see that should be expanded upon, and would you like anything like that
So just as he's coming to the microphone, the question is whether it's better to have a localized precinct for biotechnology incubator space, as it were, or elsewhere.
Thanks, Phil. It's an interesting question, I think, and there's a lot of, I guess, depending on the area of medtech or biotech that you're working on in Brisbane, there are certain different sort of, I guess, capabilities here for companies making revenue. Obviously, in the medical device space, there's a lot of companies here that Cochlear, other, especially in the in the orthopedic area as well, they've got a good basis. I think the challenge, especially space of vaccine research development and then manufacture of products, is it's a massive job, right? So it's a huge capacity and capability that you need to do that, to make products for late stage clinical or for commercialization. So the facilities you need are inherently very specialized. So high quality accredited clean room facilities that are producing significant quantities of product under a quality management system, which is set up for the purposes of either Australian research products or US or Europe, and that's demanding. But I think part of the challenge is keeping companies that get to a certain point in Australia, let alone in Brisbane and Queensland. So I think there is a gap there in the sort of mid-sized companies that are not the startup anymore. They've built up a great knowledge base. They know what they want to do, but then where do they go to next? And I think that's a gap. So that's missing in Queensland and Brisbane. You can look down to, I guess, Melbourne and companies such as Seqirus and CSL. It's more established there. So I think that's a challenge. But I think the location at North Shorter is not where we are now. That is a challenge. But to get all that capability under one roof, I think that's the most important thing. And to be able to do that in Brisbane is great. And to have Queensland government support to build that out and get it ready for when we need it to be ready. I think that's really the driver for it. That won't, I don't think it'll prevent collaboration, because we'll still need to have a presence here at the TRI, for example. We can't replicate everything that's on offer here in this building, especially the clinical trial facility, the vivarium, and also access to a community of very capable researchers. So I think it's an interesting one. I think for us, this is the best next step to sort of give us that central place where we can expand and grow. and sort of hopefully from that, and if we're successful and we do grow and expand, then other sort of companies will be able to follow suit like Microba, because you expand your growth in labs and your production capability as well. That's a good question, Ranjini. It's interesting to see the spend. Australia's interesting. I was brought up in New Zealand and spent a lot of time working in the UK. And Australia is a bit fragmented, I think, at times from a state-based approach. We're seeing investments in Victoria, New South Wales, and Queensland, the mRNA facilities, for example. Just standing on the outside looking in, you think, it's great that states are sort of proactively investing money into different technologies, but I don't know if you need sovereign protection in your state. I think it needs to be more collaborative. And I think there's some great ideas that are coming out. I think the Australian Vaccine Research Alliance, for example, trying to bring that pool together so that people to sort of to collaboratively to work and expand. For us, infrastructures, the TRI is a fantastic place for us to be right now. It's provided us with 5 years of facility in which we've been able to grow out of. And I think without it, it would have been very, very challenging for us to get to the point we're at now. I actually don't know what we would have done. And going right back to why we moved to TRI back in 2014, it was because of the clean rooms and the ability to do manufacture for phase 1 studies. And without that, we would have either needed to work with contractors or we'll try and we'll go overseas to do that work. So that was a key decision for us to come across here. And so I think access to good clinical trial centres is important. In Queensland, we're very lucky. We have that. A lot of phase 1 units here in Australia. I think then manufacturing, right? It's challenging. And the only vaccine manufacturer in Australia is CSL or Seqirus, and they can only do a certain amount in their facility. And obviously, they want something from that work and to be able to produce vaccines for the market. So I think it will be good to be able to build that up, and quite how that's done, I think, is a question probably above my pay grade, but I think there's opportunity there for engineers to do it.
Anybody else? Anyone online have a question? Chat, yes. Norbert's asking, what about bacteria biomarker in gut and skin when aiming for effective immunizing via skin patch? Did you get that?
Do you mind repeating that?
It's a very fragmented question. Norbert, is he able to ask it? No.
I think I caught.
Is there a biomarker in the gut or skin when aiming for effective immunization?
I don't think we know the answer to that question. I think there's some hypotheses formed around general upregulation of the immune system by the microbiome. And we think that's what we're seeing in the context of immune checkpoint inhibitors and other treatment response areas. But, the opportunity is to go and explore things like that. So I think it's a great question, but we don't know the answer.
And just for Angus, Alison's asking, is the Vaxxas platform adaptable to tolerizing the immune system for treatment of allergy? Alison used to work with me.
Yeah. I guess there's a number of sort of collaborations, scientists working on sort of allergy vaccination or tolerance. And we've looked at it, but we've not sort of done much work in that area. And I guess, one of the challenges there is escalating the dose of the antigen up slowly. So you obviously need a range of, and the good thing about an injectable is that you can inject more or less antigen as you try and create that tolerance. And you've probably got a bit more granularity about building up that tolerance with different dosing. So that's why kind of where we stayed away from that approach, because then you need a range of different patches or that would sort of give you that ability to cause that. I guess moving ahead to can you get allergy vaccination? I guess there's been some companies working in that space as well, and that's an area where we're interested in collaborating and with sort of novel technologies for removing sort of allergy. And I guess there's a lot of work in Australia having a peanut allergy at the moment as well as other countries.
Okay, so I think that brings us to the hour, and thank both of the speakers for a